Hello everyone. Last lecture note video for chapter 9. In our last video we talked about how if there's no oxygen available, the mitochondria shuts down. And so the electron transport chain isn't working, and so basically you're stuck with only the process of glycolysis out in the cytoplasm. When that happens, depending on what kind of cell you are, you have two different ways of dealing with it. If you are a, for example, animal body cell, a bacteria cell or a yeast cell, you might do what's called lactic acid fermentation in the absence of sufficient amount of oxygen. Some other microbes, like different kinds of yeast and bacteria, will use a different series of pathways called alcohol fermentation to deal with an anaerobic situation, which means there is not sufficient amount of oxygen available. Let's start with the lactic acid fermentation. This would be, for example, happening in your cells when they are deprived of a sufficient amount of oxygen. As usual, we start with glucose, and basically we're just drawing glycolysis here. Start with glucose out in the cytoplasm. It's broken down into two pyruvic acids. And as we've seen many times before, when we drew glycolysis, this requires an input of 2 ATPs, but it'll give you back 4 ATPs. So at least you made 2 ATPs out of glycolysis. Well, this also requires, as we've seen many times before, 2 NADs to come in here and grab some electrons and become 2 NADHs. Put a little box around that. That's basically the story of glycolysis. But in this case, if the mitochondria is shut down, the question is, what will happen to the pyruvic acid, because it cannot enter the mitochondria. Also, if the mitochondria is shut down, where is the NADH going to bring its hydrogen atom? It's full. And in order to continue to perform at least glycolysis and at least get a few ATPs out, you need empty NAD. So the NAD is looking for a place to dump off that hydrogen. Well, an extra step is added onto glycolysis here, where you could imagine the NAD coming down here, dropping off its hydrogen atom, and going back up there to strip some more electrons to continue to keep this happening. Well, that extra step that's added onto glycolysis converts the pyruvic acids into lactic acids, as you can see here. They are also 3-carbon molecules. Now, as I understand it, your body will store this, and when there's sufficient amount of oxygen left, you can convert it back into pyruvic acid, or you can somehow get those other 32 ATPs that are locked up in the chemical energy of these molecules once the mitochondria opens back up for business. Now, for other organisms, microbes, they handle a lack of oxygen a little bit different. Of course, we'll start out with glucose again. We'll break it down into 2 pyruvic acids. That requires, we put 2 ATPs in, but we'll get 4 ATPs out. We'll be able to pick up a couple of electrons here with NAD. Again, remember the mitochondria is shut down, so the NAD will come down here. There's a step added on to glycolysis, drop off its electrons, go get more, keep the ATP being made. Well, in this case, this extra step is a little different. A couple of carbons break off, one from each pyruvic acid. You make carbon dioxide here. That's the gas that'll cause your bread dough to rise, and those are the bubbles you'll see in your fermenting beer or wine. Now, this process also requires an input of a certain amount of water. And the end result are these remaining 2 carbon molecules, and that's ethyl alcohol or ethanol. And that's called alcohol fermentation. Of course, that's used in the production of beers and wines. It's also used in the making of bread, where you use yeast and the carbon dioxide bubbles are produced, causes a loaf of dough to rise and be less dense.